Exercise 17 deals with the special senses. We're going to start with the eye and then move on, talk about the ear. And the special senses basically are going to be vision, hearing, equilibrium. Equilibrium is going to involve the ear, and then taste and smell. So starting with the eye, with vision, the anatomy of the eye, most of the eye is enclosed and protected by that, the orbit, you're only seeing a small portion of the eye. It's roughly 2.5 centimeters in diameter. Starting with some of the accessory structures, you have the eyebrows, which help to protect the eyes from sunlight, and the eyelids. These are also giving protection, preventing things from getting into the eyes, the natural instinct to blink if anything comes close to the eye. The conjunctiva helps to prevent the eye from drying out. You also have the lacrimal apparatus. This helps to clean and protect the eye surface. It helps to lubricate and moisten it. And then your extrinsic eye muscles help to control the movements of the eye itself. The lacrimal apparatus, this involves the lacrimal gland, which is up in this area right here. This involves production of your tears. Now, a lot of people tend to think, oh, your tears are produced right here because this is where you see them. No. Above each eye, you have an almond-shaped lacrimal gland. It is on the superior lateral side of the eye. And there are ducts here that the tears are produced here, and every time you blink, it's going to flush those tears across the exposed surface of the eyeball. Like I said, it's helping to moisten, keep the eye from drying out. It helps to lubricate it. And the tears actually also contain certain chemicals that are bactericidal. In other words, they will break down bacteria, so it's a protective mechanism as well to prevent infection. And then as they move across the surface of the eye, like say every time you blink, then they will actually drain right here through the lacrimal canal and into the lacrimal sac, which can store the tears if you have excess production of them, and it will move down the nasal lacrimal duct and drain into the nasal cavity. I'm mentioning part of this because if you are producing tears faster than they can drain here, then it will build up here, and that's why it looks like you've got tears and may overflow and run down the front of your face. The other thing is, with the lacrimal sac here and the tears are draining in here, excuse me, if you, and unfortunately all of us have had some point in our lives where this has happened, you receive a blow right on the side of your nose right here, that basically is going to compress, and that's why your tears swell up. And the other thing is, whenever you cry, you wonder, why do you also get a runny nose? It's because where are the tears naturally draining? Into the nasal cavity. So probably much more than you want to know about crying, but that's the way it works. Your extrinsic eye muscles, as I said, there are 6 of these. They help with the movement of the eye itself and helping it to turn up and down and rotate slightly. When someone has a lazy eye, typically one of these muscles is stronger than the other, and so they can do exercises to try to help increase the strength of the muscles, strengthen the weaker side, and if necessary, they can always do surgery where they actually will shorten one of the muscles to help draw the eye back out. So it's looking where the direction it needs to be looking instead of being turned. Your photoreceptors, these are your rods and your cones. They are located inside the retina, which, so this is where the photoreceptors are located in the back of the eye. The rods allow you to see in dim light. It allows you to see black and white. It's very good for peripheral vision. The cones are better stimulated in bright light, high resolution, and they allow you to see in color. These photoreceptors are modified neurons. The rods tend to be very, very sensitive. That's why they can be stimulated in dim light. The cones, they do need to have that bright light for the activation. There's 3 different types of pigment: blue, green, and red. Like I said, that allows you to see color. If you are colorblind, that is genetically determined. And it'll determine which color you're not able to see. So what happens is the light is coming in. All of this is part of the retina. And what happens is the light comes back here is where the rods and cones are located. As you can see here in this diagram, the rods are these yellow structures, the cones are the purple ones. They become stimulated by the light itself. In turn, they're going to stimulate these bipolar cells, which in turn will now stimulate these ganglion cells. And those axons are all going to come and converge at the back of the eye and form the optic nerve, which then is going to travel to the vision area of the brain for interpretation. When you look at the eye, there are two different chambers, the lens and the ciliary zone. You'll divide it into two chambers. The posterior chamber is filled with a gel-like fluid known as the vitreous humor. This forms during embryonic development. The anterior chamber is filled with aqueous humor, and that is formed and drained continuously. So this just shows the front of the eye. Right here is the cornea. So what you see is this portion right here. This is the iris, the smooth muscle that gives the color to your eye. Once again, genetically determined. Behind that is the lens. The opening is the pupil, which is right here. So light's going to come in this way. So as I just said, the lens and the ciliary zone, this is what's the division. Back here is the posterior chamber, and that's where the vitreous humor is contained. And then the anterior segment, this chamber, front chamber, is going to have the aqueous humor, which flows this way through the pupil, through the front, and then drains out. So there's three different layers. When you look at the structure of the eye, there's the fibrous vascular and the inner of the retina, and then the humors, the aqueous humor and vitreous humor, and then the lens. So once again, if you are looking at the eye, light is coming in this way. This is front right here is the cornea. Here is the pupil. Here is the iris. Here is your lens. Back here is the posterior segment filled with that vitreous humor. This yellow layer right here is the retina, where we just talked about. I showed you where you've got your rods and your cones, the bipolar cells, the ganglion cells. Behind that is the choroid layer. Behind that is the scalar layer. And this is showing the optic nerve. With the optic nerve leaving the back of the eye and going to the visual cortex, you also, along with that, will have your blood vessels that are coming in and supplying all of this tissue. So the fibrous layer is that outermost one. The scalar is the posterior portion. This is what's referred to oftentimes as the white of the eye. The cornea is a small portion in the anterior. It's transparent to allow light to enter the eye. It does have nerve endings, but there's no blood supply to the cornea. That is why you can do a transplant of the cornea and don't have to worry about rejection issues, is because there is no blood supply to it. The vascular layer is the middle layer. It's pigmented. There's three regions to it. You've got the choroid in the posterior section, the ciliary body, and the iris. So the choroid has a very rich blood supply. It's pigmented to help absorb the light. And there is that opening where the optic nerve will leave in the back of the eye. The ciliary body does encircle the lens. It helps to hold the lens in place. It helps to control the shape of the lens, because the lens will change its shape depending on what you're looking at, because it needs to focus the image back on the back of the eye on the retina. The iris is that colored portion. In the middle is the pupil, which is actually a hole that allows the light to enter the eye. The iris is a smooth muscle, so you do not have control over it. When it constricts, Basically, it's usually with very close vision and in very bright light, so it's decreasing the amount of light coming in. When it dilates, that usually happens in dim light to allow more light to. The retina is the innermost layer. It does contain the photoreceptors, the neurons, and glial cells. It has two layers, a pigmented layer, which is the outermost one, and then the neural layer, which is the innermost of the layers. So that pigmented layer, it's a single cell layer. It helps to absorb light and prevents it from scattering all over the place. It also does contain phagocytes. This is important because the pupil is an opening to the eye. You do not want take a risk or take a chance excuse me, of having any foreign substance such as bacteria enter the eye through the pupil, be able to travel through that vitreous humor, and then from there travel via the optic nerves and enter the brain. And so if you have phagocytes, what they do is they are able to recognize anything that's a foreign cell, any foreign entity, and they will engulf it. So it's a protective mechanism. This pigmented layer also stores vitamin A, which is going to assist with the overall process of helping you to receive the impulse and basically to see better. The neural layer is composed of these photoreceptors, the bipolar cells, those ganglion cells. The optic disc, that's where the optic nerve leaves the eye because there's no photoreceptors right in that spot. It's referred to as the blind spot. So light's going to activate those photoreceptors, the rods and cones, which as I said then stimulate the bipolar cells, which then stimulate the ganglion cells, and all those axons from those ganglion cells, if you remember from that previous picture I showed you earlier, will then all come and congregate and leave the eye at the optic nerve. The lens is biconvex, it's transparent, it is avascular. It should be very flexible. Now, sometimes as you age, it becomes less flexible, but in a healthy young adult, it should be very flexible. It's going to focus the light on the retina, and it's held in place by the ciliary zonule. Excuse me. You'll notice in this diagram is showing with the right eye, and that blue is showing the field of view, the vision field of view, and then the left eye is shown in yellow. And what is going to happen is that image that you're looking at, the lens is going to focus that image on the back of the retina. And so it's going to focus it right back here, in the back of the eye on the retina, so that the photoreceptors are stimulated. And then what happens is that image is then going to move via the optic nerves. It's going to come back here, and in the optic chasm, half the nerves go this direction. There's a crossing over that occurs from each eye, and then in the visual cortex area of the brain, you're going to interpret what you're seeing, and you're able to overlap these areas. The brain knows to overlap them, and that's how you get depth perception. You put the two images together. Now, this is showing a dissection of a sheep eye. A couple things I just want to point out. You've got the sclera, here's the retina. Now they've pulled aside the retina. It looks like just a thin membrane. And sheep, as well as some other animals, have what's known as the tapetum lucidum. It's a very pretty kind of a pearlesque type of, very pretty blue. This helps, basically what happens is the light comes through, it hits that, and it's going to reflect back to the retina to increase the chances of stimulating those photoreceptors, and that's what gives some animals very good night vision. As pretty as it looks, humans don't have this, but it's really pretty to see if you get a chance to do an actual eye dissection. Down here, this gel-like substance is the vitreous humor. Back here you can see the retina. It's a very thin membrane. This is the lens right here. And so if you were to remove the lens, you would actually see a hole, which would be the pupil. For hearing, the two special senses are equilibrium and hearing that are associated with the ear. For hearing, if we look at this, it's going to involve all components of the ear, the external ear, the middle ear, and the inner ear. For equilibrium, it's just going to involve the inner ear. So the external ear is going to be composed of the auricle or the pinna. This is what most people think of when you see that external part of the ear. Its purpose is to help funnel the sound waves into your external acoustic means. And this is also known as your auditory canal. That's going to extend down to the eardrum. So you're trying to help focus and direct, essentially, the sound waves through the auditory canal to the eardrum, which is also known as your tympanic membrane. That is the division between your external ear and the middle ear. And what that's going to do is start vibrating in response to those sound waves. So here is your external ear right here. And then right here is the middle ear, which contains the auditory ossicles, or the bones. And then this portion right here is the inner ear. So the external ear, as I said, this is going to help direct those sound waves in the auditory canal till you hit this light color right here is your tympanic membrane. It is enlarged over here so you can see, or the eardrum. So it's going to direct those sound waves in. And then in the middle ear here, it's going to get, the eardrum starts vibrating. And then that's going to get these bones vibrating. So each one successfully stimulates the next bone to get it vibrating until it vibrates here along the round window. And that's going to amplify the sounds. This should be filled, the middle ear should be filled under normal, healthy conditions with air, not fluid. So I said it's going to transmit those vibrations from the eardrum to the auditory ossicles, which amplifies the sound. Those auditory ossicles are known as the malleus, the incus, and the stapes. And this is shown right here as the order that they are in. And then the internal ear, this is also going to be where the receptors are, the sensory receptors for hearing, that then will, the sound waves have been amplified, they will be stimulated, and then they will send the electrical impulse to the auditory section of the brain for interpretation of that sound. The internal ear, you have the bony labyrinth. This contains the perilymph. This includes your vestibule, which is your sacral and the urticle, the semicircular canals, and the cochlea. Now, the hearing receptors are actually in the cochlea. And then you also have the membranous labyrinth, which contains endolymph. So this is showing here with the inner ear, your semicircular canals here, and then the cochlea here. This is where the receptors are going to be. The semicircular canals are going to be more involved with equilibrium. And this is showing the anatomy of the cochlea, where internally you do have these hairs, and that's where you're going to have your receptors that pick up the stimulus for essentially now hearing and send it via the nerves. So the sound waves get funneled in, the tympanic membrane is going to vibrate, the ossicles will amplify those vibrations, they cause the oval window to vibrate, and then those pressure waves then move through the fluid that's there in the cochlear duct, and then it's going to cause the vascular membrane to vibrate, and that stimulates those hairs on those inner hair cells. And then that will generate the impulse. So this is just showing how you have the sound waves and the pressure, and it gets amplified in the middle ear. So each auditory cortex receives impulses from both of the ears. The tones perceived separately. It can detect the pitch, the localization of it, the intensity of it. Hearing does generally decline as we age. Equilibrium. What's going to happen is in the semicircular canal, there are receptors that can help monitor different rotations or positions of the head. And these receptors in the semicircular canals, known as the vestibular apparatus, are going to help not only regulate and monitor the head position, but also any type of linear acceleration. Where are you positioned relative to everything else around you? And so for equilibrium, it's not just these vestibular receptors that are detecting from the inner ear. It's also, you have visual receptors, and then somatic receptors, receptors that are monitoring at your joints. Are you leaning forward? Are you leaning towards the side? And like I said, visual receptors, are things spinning? Are they turning? All of this information is coming to different areas in the cerebellum and in the cerebrum that's monitoring in the brain all of this incoming information, and together then it's monitoring. Are you upright? Are you about to fall over? Are you spinning? Etc. And that all works together and plays with your equilibrium. In terms of chemical senses, this involves smell, which is known as olfaction, and taste, which is known as gustation. So chemical receptors are going to be responding to chemicals, usually in an aqueous solution, at least for humans in an aqueous solution. Olfaction, the sense of smell, the receptors are going to be located in the roof of the nasal cavity. When you sniff, where you're bringing in more air and it's able to swirl around more in the nasal cavity, increasing the chances of stimulating these receptors. The olfaction receptors are going to be bipolar neurons. They, in order to be stimulate, they must be in a gas phase entering the nasal cavity, and then they're going to be dissolved for the receptor to be stimulated. There's a very wide range of sensitivity for the olfactory neurons, and this tends to decline as we age. So this is just showing how when you sniff, you're going to bring in this air. It's going to swirl around in that nasal cavity, and up on the roof of the nasal cavity is where the sensory receptors are actually located. What's going to happen is the olfactory receptors will become activated, since it involves the olfactory bulb. And then, as you can see, it's going to go through a series of steps from there to the mitral cells, olfactory tract, olfactory cortex. It's all going to go through the limbic system. And remember, the limbic system involves emotion, so that is why you put an emotional connotation on smells, be it good, bad, or indifferent. You may think of certain smells and have this emotional trigger for it. Why? Because it goes through the limbic system. And this is just another example of showing how you have the stepwise process of triggering these receptors to then eventually send that impulse to the brain. Gustation, this refers to tasting. Your taste buds are going to be the sensory organ. Most of these are located on the tongue. The food must be dissolved to stimulate them because they are located below the surface of the tongue. So they must be dissolved. They must be in liquid form to stimulate it. Saliva, if you're chewing something solid, saliva is going to dissolve it and get it into that liquid form so you can pick up the taste sensation. The 5 main taste sensations are sweet, sour, salty, bitter, and umami. So the nerves for taste are also going to go through the limbic system, so that's why you also have an emotional context given to taste. The other thing is taste is been estimated to be 80% of it is actually smell. I would challenge you as to whether or not you could describe to someone what a food item tastes like without thinking of the smell. So if you were to describe to someone what barbecued pulled pork taste like, or a nice juicy steak, or crawfish boil? What do those food items taste like? If you had to describe it to someone who's never had it before, could you do it without in your mind thinking of the smell of it? Can you describe to someone what a nice crawfish boil tastes like without thinking of the smell of cayenne pepper or crab boil? And you probably can't do it because taste and smell are so intertwined and they both go through the limbic system. Once again, you're going to put some type of an emotional connotation with it. This is showing on the tongue the different areas of where the taste buds for the sweet versus salty. Bitter, as you notice, is on the back of the tongue. This is a survival mechanism. A lot of poisons, natural poisons, have a bitter taste to it. And so that's why it's on the back of the tongue and often triggers that gag reflex to prevent you, from a survival mechanism, prevents you from eating or drinking something that may be a poison. But you'll notice that the taste buds themselves are below the surface. So you have these indentations, and it's down here where the taste bulbs are. That's why it has to be in liquid form for you to taste.